I miss y'all. I know I've said it and I'll keep saying it, but I really do. So first off, you notice I'm in the new classroom, which I wish y'all were here with me because it's not nearly as much fun without you. But I did want to show you the new building while I'm here. So we have our door and our supply cart. You have your calendar. I did sneak the Christmas lights in, Promethean board, and then I'll just kind of slowly pan. We have our like supply station and stuff right there, and I'll just slowly pan so that y'all can see the room. There's your review on my computer, which we're about to go over. So, anyway, that's the room. I can't wait for y'all to be here with me. Until then, we're gonna go over our review for the test on Monday. So, first thing to note, we're just gonna start kind of where we did with the review and the beginning of the unit, which is we talked about different cell processes, different cell cycles. So now we are on to mitosis and meiosis, which are the two different types of cell division. So we're gonna start with the simple one, mitosis, which is your asexual reproduction, which is gonna produce your genetically identical cells with the same number, 46 chromosomes. And so mitosis is gonna happen through one set of 4 stages, which is going to be your prophase, metaphase, anaphase, telophase. Remember, we said prophase is where all of our chromosomes are going to condense down. Metaphase, they line up in the middle. Anaphase, they're starting to pull apart. And telophase, it forms those 2 different cells. And so, that being said, like we said, same number of chromosomes, 46, genetically identical. Starts getting a little bit trickier when we then get into our meiosis, which is our sexual reproduction, which is where that genetic variation comes in. So you still go through that prophase, metaphase, anaphase, and telophase, but this time we do it twice, and that purpose is that through doing it twice, it then cuts down the number of chromosomes. So we go all the way through that twice, so that we end up having 4 cells that are genetically different, but they are haploid, meaning they have half the number of chromosomes, so 23. And that is gonna provide that genetic variation, which is so important. And so looking at all of this, specifically with meiosis, that genetic variation is gonna come from one specific thing, which is called crossing over, which we said occurs in prophase 1. So that's going to happen. You have prophase 1, the genetic variation occurs, and you cross over. Then you have metaphase 1, anaphase 1, telophase 1. Then we regroup. At this point, we have 2 cells that are genetically different with 46 chromosomes. And then both of those cells go through meiosis 2, so prophase 2, metaphase 2, anaphase 2, telophase 2, which is gonna break us down to those 4 cells that have 23 chromosomes each. So then looking at that, all of this is happening inside our nucleus. Oh, and they're testing. I'm just gonna keep going, and I hope that y'all can hear me. So inside the nucleus, we have our chromosomes. Inside the chromosomes, those are made up of our DNA, which are made up of our genes. And the genes, we said, were the combination of those 4 different bases. You have your thymine, guanine, adenine, and cytosine, which you're not required to know, just for your information. So chromosomes are going to contain genetic information, genes, alleles, and DNA. All of those are different ways of saying the same thing. All of this was studied and perfected by a man named Gregor Mendel, who started looking at the different traits of pea plants and wondered how those came about because they look different. And so he started studying those traits, which is known as genetics, or the study of heredity. Whereas heredity means the actual passing on of those traits, with traits being physical characteristics. So we said that Mendel started looking at them, and he took 2 different parents, one homozygous dominant and one homozygous recessive, and crossed them, which gave us all heterozygous offspring. And then he crossed the heterozygous against each other, and that gave us that really typical 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive pattern that we see within Punnett squares. And so all of this happened because of those different alleles, which are your different traits. So we said you have a trait, which might be eye color. However, your different alleles would be like brown eyes versus blue eyes, or something like that, where brown eyes is going to be dominant over that. So what does dominant mean? Dominant means that you have a trait that is always going to show up if the dominant allele is present. And then recessive only shows up if there is a lack of that dominant presence. So you have different ways that all of those can then combine. You have homozygous dominant, which is where you're going to have two dominant, which is going to be represented by two capital letters. You have heterozygous, where you have two different, so you have a dominant and a recessive, which is going to be a capital letter and a lowercase letter. And then you have homozygous recessive, which is going to be two different recessive, or two recessive alleles that are the same, which are represented by two lowercase letters. And so that is when we start getting into Punnett squares. So hold on, I'm going to take you guys over here. And I'm going to go ahead and do a couple Punnett squares on the board for you so that you can kind of follow along and see where I'm going and what I'm doing and where my thought process is. So we said freckles are dominant to no freckles. Perform a Punnett square cross between two heterozygous parents and find the percentages of phenotype and genotype of the offspring. Can any of them pass on I meant to say recessive trait, so can any of them pass on the recessive trait to their children? So we're going to look at that and we're going to say we have, let's see if y'all can see this. Kind of, okay. It's a little glary, so we're going to take that down so that you can see it. So that being said, we have, we said two heterozygous parents. So because they're the same, it doesn't matter. Our mom is heterozygous, so capital and lowercase, and our dad is heterozygous, so capital and lowercase. So the first thing to remember with Punnett squares is we have 1, 2, 3, 4 boxes, which means each of the boxes are going to be worth how much? Okay, feels a little Dora the Explorer because I know that y'all can't actually answer me back, but they're worth 25%, which I know that y'all all know. So you're going to go to the top left box first, and you're going to say, okay, my left one up here is a dominant, and then here, my top one is a dominant. So for this one, I'm going to get capital, so I'm going to have homozygous dominant. Then for my upper right box, I'm going to have a capital dominant from over here and a recessive from up here. So that is going to be capital lowercase, so heterozygous. Then here, same thing, I'm going to have a capital from up here and a lowercase from here, which is going to give me a heterozygous. And then here, I'm going to have a lowercase and a lowercase, which is going to give me homozygous recessive. So all of that being said, up here, I have one homozygous dominant, two heterozygous, one homozygous recessive. So I'm going to have 25% that are those homozygous dominant. So I look at that and I say, okay, 25% homozygous dominant, does that mean that they're going to have freckles? Yes. So genotype here is going to be the genes, which is homozygous dominant. Our phenotype is going to be the fact that they have freckles. And then looking at that, can a homozygous dominant person pass on the recessive trait? No, they don't have the recessive trait to pass on, so they will not pass that on. Looking here, we have two heterozygous, which is going to add up to be 50% heterozygous. Again, heterozygous has the dominant trait, which means automatically the dominant trait is going to show up. So heterozygous is going to have freckles. And then looking at this, capital and lowercase, can they pass on a lowercase gene? Yes. They'll pass it on 50% of the time because they only have one of them. So yes, 50%. Then looking here, we have one that is homozygous recessive. So 25% homozygous recessive. Will they have freckles? No, no freckles because they don't have the dominant gene. And they will pass on the recessive 100% of the time. So let's see if you can see that well. You can see that fairly well. So we have that homozygous dominant, heterozygous, homozygous, recessive area there. So the next one, we said straight hair is dominant to curly hair. Perform a Punnett square cross between a homozygous recessive dad and a heterozygous mom. What is the percentages of phenotype and genotype? Genotype is the arrangement of the alleles. Phenotype is the physical presentation. And can any of them pass on the trait to their children? So we're going to look at this a little bit differently. We said we have a homozygous recessive dad, so we're going to have 2 lowercase here, which means he's going to have curly hair. And then we have a heterozygous mom, which means that she will have straight hair. So as I start to look at those different crosses, I'm going to take my upper left-hand box here, and I'm going to say that this has a capital and a lowercase. So this one is going to be heterozygous. Here, I have a capital, dominant, and a lowercase recessive. So this one would be heterozygous. Here, I have a lowercase and a lowercase. So that is going to be homozygous recessive. And then here, I have a lowercase and a lowercase. And so that is going to be homozygous recessive. So I look at this and I say, okay, I have 2 that are heterozygous, so that would be 50% heterozygous, which means that they have straight hair because it is dominant. And then can they pass on the trait? Yes, they will pass on the recessive trait 50% of the time. So we put that. Then we have 2 that are homozygous recessive. So 50% are going to be homozygous recessive, which means they're going to have curly hair, and they will pass on that trait 100% of the time. So that is how that's going. And then the last one, we said that we have, I believe, a homozygous dominant and a homozygous recessive, but let me check really quick. Dimples are dominant to no dimples. Perform a Punnett square cross between a homozygous dominant dad and a homozygous recessive mom. Answering those the same questions. So, dad is going to be on top. We said that he is homozygous dominant for dimples, and then mom is homozygous recessive for dimples. So, we are going to bring down our dominant from our dad and our recessive from our mom here to form heterozygous. Then dominant from our dad, recessive from our mom for heterozygous. Dominant from our dad, recessive from our mom for heterozygous. And then dominant from our dad and recessive from our mom for heterozygous. So we look at this one and we see that all of offspring are exactly the same, which means we have 100% heterozygous, so capital and lowercase, which is going to be that all of them have dimples, and all of them can pass on the trait 50% of the time. So, again, kind of going over Punnett squares. They're not as hard as you think they are. If you have any questions, please reach out. Let me know. Ask. I can always do more videos. So if you have any questions, just let me know. I know y'all will do awesome. There's a completed copy of the review in my handwriting posted, and there is a review game posted so that y'all can go on to Kahoot and play with yourself, play with friends, whatever you want. But anyway, I miss you guys. I love you. I'm praying for you, and I can't wait to see y'all on April 3rd.